Hemoglobin. Hemoglobin is a tetramer consisting of two dimers, 2 alpha and 2 beta. The variant forms which cause disease, these hemoglobin variant forms are mutant forms of hemoglobin caused by variation in genetics. This is a structure, 3D structure of the hemoglobin. The hemoglobin S is a variant from hemoglobin found in people with sickle cell disease. There is a variation in the beta chain gene causing a change in the properties of hemoglobin, resulting in sickling of red blood cells. While the hemoglobin C form is another variant due to the variation of the beta chain gene. This variant causes a mild chronic hemolytic anemia. Other variants cause no detectable pathology and are thus considered non-pathological variants. Hemoglobin A1C actually is called glycated hemoglobin. To a small extent, hemoglobin A slowly combines with glucose at a certain location in the molecule. The resulting molecule is often referred to as hemoglobin A1C. As the concentration of glucose in the blood increase, the percentage of this variant will be this form of HB hemoglobin A that turns to HB1C increases because it binds with glucose. In diabetics whose glucose is usually high, the percent of HbA1c also increase. Because of the combination of A with the glucose, the A1C percentage is representative of the glucose level in the blood over the past 2-3 months. So usually patients who have diabetes, they do analysis for this type of hemoglobin to see how they are managing the diabetes. You can see from the figure, for people whose diabetes, without diabetes, the normal range of hemoglobin for A1C is between 4 to 5.6%, while if the level between is the level between 5.7% to 6.4, that means they have a higher chance of getting diabetes. Levels of 6.5% or higher means you have a diabetes. The hemoglobin concentration. So actually you can weight of the hemoglobin, it's a weight of hemoglobin per unit volume of blood, gram per deciliter, gram percent, gram per liter. The increase in heavy smokers, high white blood cells counts, there's a false increase and due to dehydration. While it decreases in pregnancy, children resting from upright position and while taking IV fluid. And of course the procedure is only for knowledge. The hemocrit or the packed the cell volume. You can see that it is the ratio in percent of packed red cells in total whole blood in percentage. The reference value for adults, for male it's 40 to 54%, while female is 37 to 47%. So you can see from the figure it's the packed volume of the red blood cells, and as well the procedures is for knowledge. The example, if the column of the red blood cells measure around 20 millimeter and the whole blood column measures around 50 millimeter, then the hematocrit will be 20 over 50, 0.4 or 40%. Normally we have an automated cell counter that calculates the hematocrit by multiplying the red blood cells number in millions per millimeter cube by the mean cell volume in femtoliter. We'll talk now about blood indices. The first one is the mean corpuscular volume. It's the average size of RBCs. And the equation, you don't need to be familiar with the equation. The reference value from 80 to 96 florins per red blood cells in adult. They increased in macrocytic anemia, infants, newborns, and chronic alcoholism. They falsely increased in marked leukocytosis, hyperglycemia, and reticulocytosis. They decreased in microcytic anemia. The mean corpuscular hemoglobin, it is the quantity or weight of hemoglobin in average red cells. Actually, so it's a hemoglobin over red blood cells. The reference value, it's around 27 to 33 picograms per cell in adult. The increase in macrocytic anemia, infants and newborns, and decreased in microcytic anemia. The mean corpuscular hemoglobin concentration. Actually, it's the hemoglobin concentration divided by the BCV. it is a concentration of hemoglobin in the average red blood cells. The reference value are 33, 36 gram per deciliter in adult. They increased in hyperchromic anemia, decreased only in hereditary spherocytosis, more than 36 milligram percent. The normochromic or normocytic anemia, it is normal-sized RBCs but a low number of them. Normal-sized RBCs but a low number of them. So with elevated reticulocytosis, that means immature RBCs in the blood, there are normo-regenerated anemia. This result of a normo-regenerative anemias such as immunohemolytic anemias, glucose 6-phosphate dehydrogenase deficiency, hemoglobin S or C, and some other example hereditary spherocytosis. With low or normal reticulocytosis, reach percentage from 0.5% to 2.5% in adults, hyporegenerative normatic anemias such as chronic diseases, chronic renal failure, and aplastic anemias. Decreased MCV and decreased MCHC. These can be a result due to microcytic hypochromic anemia, like iron deficiency, thalassemia, anemia of chronic disease, lead poisoning. While the other way around, if MCV is increased, that's mean a macrocytic anemia or normochromic anemia, like that may result from folate deficiency, B12 deficiency, hypothyroidism. The ESR, erythrocytic sedimentation rate, these are the normal values for adult and the children's. You don't need to memorize these normal values. It's only useful for diagnostic 3 diseases: myeloma, temporal arthritis, and polymyalgia rheumatica, in which it may exceed 100 millimeter per hour. From this slide, which is only for knowledge, you can see how the example of red blood cell indices with corresponding red blood cell counts and everything, and MCAV and MCH and MCHC. So you can see how they interpret the result to link it to a certain abnormality or disease. And this slide is only for knowledge. The red blood cell distribution WADS. So it's an indication of the variation in the RBC size. It is derived from the RBC histogram, like shown in the figure. In general, an elevated red RDW indicates more variation in the cell size, has been associated with anemias with various deficiencies such as iron, B12, or folate. Thalassemia is a microcytic anemia that characteristically has a normal RDW. So you can see from the figure, how can you calculate the RDW? It's the standard deviation of MCV times 100 over the MCV. The normal value is around 11.5 to 14.5%. The increased value indicate ansiocytosis. So you can see here the cell size on the x-axis and the cell number on the y-axis. So actually this figure distributes the different sizes of RBCs. As an example, you can see here, if you have low ansiocytosis, the variation of size is a bit lower, appearing as well from the figure below. While if you have high aniocytosis, you can see that the variation of sizes is a bit large. So you can see that the curve in the below figure is quite spread. So here the RDW is around 16%, while here it's around 13%. So this one represents normal MCV with high RDW. It's a case of anisocytosis. This one represents that low MC value is shifted to the right, to the left, while this one represents dimorphic RBC population, that you have generate 2 major population with 2 different sizes in the red blood cell distribution. Now we'll talk about the white blood cells, leukocytes. These are cells of the immune system which defend the body against both infection, disease, and foreign materials. A number of white blood cells per unit volume are expressed in these units. is often, and their number is often an indicator of disease. They are normally between these number of white blood cells in a liter of blood, making approximately like 1% of the blood. Generally, they are classified as 5 types of white blood cells. The high number of white blood cells, leukocytes, may indicate infectious disease such as bacterial infection, inflammation such as appendicitis, leukemias, various types of leukemias, hemolytic disease of newborn, uricemia like gout disease, ulcers, severe emotional or physical stress, tissue damage, for example burns, as well as leukopenia, viral infection like AIDS, thyroid fever, and infectious hepatitis. While the high number of white blood cells, leukocytosis, may indicate cirrhosis, which is a chronic disease of the liver, characterized by the replacement of the normal tissue with fibrous tissue with no function, so the liver loses its function, and this due to alcohol abuse, nutritional deprivation, and the infection by viruses. As well, high numbers may indicate bone marrow failure due to tumor or fibrosis. As well, presence of cytotoxic substance, collagen vascular disease such as lupus, disease of the liver or spleen, radiation exposure, and the drug therapy. The white blood cells count, of course, the procedure is only for knowledge, and as we said before, you use the same hemocytometer. You count the total number of squares on the periphery from 1 to 4 and then multiply to 50.